All right, today we are going to continue on our discussion about the cell cycle and cell division by looking at how it's regulated. So, how does a cell know when to divide? How does a cell know when it shouldn't? How does a cell know that if it is surrounded by a lot of other cells, it probably shouldn't make another cell and take up even more space? All of these things are regulated in a deep and a complicated way. And we're going to just take a look at a very superficial look at how that's accomplished today. So a multicellular organism needs to coordinate cell division across coordinate cell division across different tissues and organs. And that's really critical for normal growth and development. So no two maybe tissues would need their cells to divide at the exact same time or at the exact same rate. Not all cells can have the same cell cycle. So that what's telling each type, like a skin cell versus a brain cell versus a muscle cell versus a lung cell, when and if and how fast to divide. So the frequency of cells dividing varies by the cell type. Embryos will go through the cell cycle once in less than once and one time less than every 20 minutes. So every 20 minutes, a new cell will divide in really early embryos. They're dividing super fast. In skin cells, they divide really frequently throughout their life. So every 12 to 24 hours, a given skin cell will divide. Like we talked about, liver cells that exit and go into G0 phase retain the ability to divide, but keep it in reserve only when some injury occurs. So they might divide only once per year or two. And then there are others like mature nerve and muscle cells that do not divide at all and are permanently in G0. So how can all these different timings be regulated and in a way where it allows the organism to maintain homeostasis? Now, we're going to talk about how this is controlled by checkpoints in the cell cycle. And from our talk last time in cell cycle, there are really two irreversible points in the cell cycle where something really important happens where they can't go back. And that is one in the S phase, the replication of genetic material. Once a new copy of DNA has been made in preparation for cell division, you really can't go back. You can't destroy half the DNA and now only have one copy. So that's kind of like a one-way street. There's no coming back from this type of point is the replication of genetic material in the S phase. The other is the separation of sister chromatids moving into anaphase from metaphase. So once sister chromatids have separated and now they are independent chromosomes going to the opposite poles of the cell, you can't glue them back together. Though it's done is done and there's no going back. So these are used, amongst others, as checkpoints where the cell will pause its movement through the cell cycle and process and assess if everything is as it should be to move on. So, for example, there is one of these checkpoints in metaphase that makes sure that each, or sorry, there's one of these checkpoints, for example, in the S phase, where there's a pause in the progression through the cell cycle, where before the cell exits the S phase into G2 phase, um, the cell stops and looks at each chromosome and makes sure that it's double-stranded, it's been duplicated correctly, and has two sister chromatids. So we check to make sure that happens. That's okay. Okay, cool. And then later, as I was saying, earlier that there's a spot at the end of my metaphase right before anaphase where the cell halts and makes sure that there's a spindle fiber attached to both sides of each chromosome to ensure that they separate properly in anaphase. And once that is ensured, cell can go ahead and go through um, into anaphase without any fear of um, an incorrect number of chromosomes in either of the cells. So these are examples. That was an example of two things called points called checkpoints. And these checkpoints are what regulate the cell cycle. These checkpoints are stop and go chemical signals at critical points. And really, there are three key points. There's a G1 checkpoint, a G2 checkpoint, and an M phase checkpoint. And the cell will naturally, as it progresses through the cell cycle, stop at these checkpoints unless it receives a signal, a chemical signal that says, yep, everything's okay. You can go past this point. And those signals are only produced if key cellular processes, such as replicating DNA correctly or spindle fibers being attached to every centomere correctly, um, are what keeps the cell in a stop situation at these checkpoints and unable to go on. So, as I said, there are three major checkpoints in the cell cycle. There are many other smaller ones, but we will be most want to be familiar with these three. There's a G1/S checkpoint right here, right before the cell exits the G1 phase into the S phase. And it's basically checking, hey, can DNA synthesis begin? Do we all have all of the materials and resources we need to go through and replicate DNA? If the signal says yes, that checkpoint will be moved past and DNA synthesis will occur. There is then the G2/M checkpoint right after G2, right before the mitotic phases, and the cell will stop here and assess, has DNA been synthesized correctly? Do I have, again, enough energy to go through with mitosis? If not, I will wait here, correct any errors that occurred in DNA synthesis, gather more resources, and then move past into mitosis. Finally, in metaphase of mitosis, as we discussed previously, there's a spindle checkpoint where the cell pauses momentarily in metaphase to see, are all chromosomes attached properly to spindle fibers, and can the sister chromatids separate correctly? If not, it will wait until that occurs, and if yes, it will move on and divide and go through anaphase and telophase and cytokinesis. So these checkpoints control when a cell is able to move past certain points. Of all of them, the G1/S checkpoint is the most critical. It's the primary decision point or the real restriction point. If a cell receives the go signal at this point, it will divide. It might be a matter of time, but it will, right? This cell's internal signals, like its size and nutrition, as well as external signals, like growth factors, are all important in allowing or prompting the cell to move past the G1 checkpoint. If the cell does not receive signals externally and internally to move past the G1 checkpoint, it will exit the cell cycle and switch into G0 phase. So things like neurons and muscle cells never receive or are incapable of receiving that signal, and that's why they stay in G0 phase their whole life upon maturity. As we talked about, that G0 phase is a non-dividing differentiated state where actually at any given time most human cells are in. Liver cells, for example, are one of the only ones that can go in and out based on damage and need, but others like nerve and muscle cells that are highly specialized always stay in this G0 resting phase and never divide. They never get the call back. So that's great, but how do cells know? What are these signals? How do they know when to divide? Well, there are chemical signals in the cytoplasm that give the cell a cue. And these signaling molecules are usually proteins, activators that can encourage the cell to move past these checkpoints, and inhibitors that can kind of prevent these cells from moving past these checkpoints. The main chemical signal we need to be aware of that has to do with progression past checkpoints is called cyclins and cyclin-dependent kinases. These are internal signals or promoting factors, not to be confused with external signals that a cell might get to go through division called growth factors. And just like if you remember our signal transduction pathways, our phosphorylation cascade, the primary mechanism of controlling the on signal and the off signal or go through the cell cycle, go past this checkpoint is phosphorylation. Phosphorylation. So that's why we have again these kinase enzymes, cyclin-dependent kinases. It's a kinase, that means it's going to phosphorylate something, and it phosphorylating something will help the cell move past its checkpoint. So this is the role of cyclin-dependent kinases and cyclins in moving a cell through the cell checkpoints. Okay. Now, CDKs or cyclin-dependent kinases are always present in the cell. Cyclin-dependent kinases are always present um, in the cell, and um, they are proteins that kinase that phosphorylate cyclins. Cyclins are proteins that have a regulatory effect on the cell cycle. As the cyclin concentration increases in the cell, or the cyclin concentration is not always constant in the cell, like cyclin-dependent kinases is, but rather it grows and it shrinks throughout the cell cycle. Now, as more cyclin accumulates in the cell, the cyclin-dependent kinases are able to join with them and phosphorylate them in what's called a cyclin-dependent kinase-cyclin complex. And as the complex of cyclin-dependent kinase and cyclin increase, as the quantity of the CDK cyclin complex increases, the cell is pushed farther and farther along. Um, the cell cyclin passes certain checkpoints. So this here is a picture of an activated cyclin-dependent kinase. It's phosphorylated and is joining with this cyclin to form a CDK cyclin complex. And it's the concentration of the CDK cyclin complex that pushes the cell through the cell cycle. So here's a picture. Here's a picture of the cyclin concentration. And MPF, or maturation promoting factor, that's another name for the CDK cyclin complex. So you can see as cyclin increases through the cell cycle, at its critical, its highest point is when it triggers moving past that spindle checkpoint, and then upon that, its concentration rapidly decreases to where it's at its lowest right before G1 phase, and then it will gradually grow again and shrink, and gradually grow again and shrink. And this growing and shrinking, the growing is due to the concentration of cyclin dependent kinase in cyclin interaction increasing, and so there's more and more and more and more active CDK cyclin complexes, and then after a critical value, a critical point, right here where it reaches its peak at the spindle checkpoint, the protein phosphatases will dephosphorylate the complex, and then their concentration just plummets, and it all starts over again. So here is our cell cycle, and it uh, shows a number of different regulatory elements in our different checkpoints. So here is our G1 checkpoint, that growth factors and nutritional states of the cell and the size of the cell all influence the CDK and cyclin levels. Here at the M checkpoint, the replication completing in the DNA integrity all affect the CDK and the cyclin levels. And here is the spindle checkpoint where chromosomes attached to the metaphase plate uh, activate or inactivate what's called mitosis promoting factor and anaphase promoting factor to allow the cell to pass into anaphase. So the CDKs and the cyclins drive cells from one phase to the next in the cell. Proper regulation of the cell cycle is so key to life that the genes for these regulatory proteins are highly conserved throughout evolution. They're very, very similar in every form of life that we know of. The genes are basically the same in yeast, insects, plants, animals, and us and humans. So they're very, very old, developed a long time ago because every cell, every organism does it pretty much the same way, regulating their cell cycle, that is. Um, we also talked about how external factors can, like called growth factors, can promote or inhibit cell division. Proteins signaling the release by body cells that stimulate others to divide. So if there are holes or empty damaged tissue, those cells that are near that damaged tissue will release some growth factors to induce others nearby to grow and divide to fill the void. And these cells will stop secreting this growth factor when they become surrounded by other cells. And remember that juxtacrine signaling, they'll feel the receptors, the protein markers on each other and know, okay, I'm packed in here close. I don't need to divide anymore. So most body cells, most normal body cells display what's called density-dependent inhibition. When the cells are crowded, they stop dividing, and each cell binds a bit of growth factor, and there's not enough activator left to trigger division in any one cell. And they also exhibit anchorage dependence. So to divide, cells must be attached to some sort of substrate, some sort of network of other cells. And this is kind of like you might think a touch sensor that, hey, I'm with all my other cells, so I'm attached to something. They can't just go freely floating through the blood and start dividing, and making a big clot or something. Here is a picture of maybe a growth factor in a cell surface receptor through a phosphorylation cascade, causing an increase in cyclin-dependent kinase cyclin concentration, which then could promote cell division. Some examples of growth factor are like platelet-derived growth factor or PDGF. This is made by platelets in the blood um, uh, when a clot is needed. So when there is an injury or a tear in a blood vessel, platelets will start to release platelet-derived growth factor, which will, when a platelet binds to it, will stimulate the cell division in fibroblasts and connective tissue to start to heal the wounds. So the release of this platelet-derived growth factor will cause new cell division in fibroblasts and allow connective tissue to start to grow and to heal the injured area. Now, growth factors can also lead to cancer, and cancer is one of the big results of the cell cycle regulation gone wrong, when the cell cycle is failed to be regulated. So there are uh, genes that encode growth factors called proto-oncogenes. And these proto-oncogenes are normal growth factor genes that become cancerous when they're mutated. So they normally produce growth factors, but if a mutation would occur, they might um, they might start to go haywire and permanently stick a switch on or permanently cause high cyclin levels in the cell. And so the cell will be continuously stimulated to divide faster, 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 and form, for example, a tumor. Um, RAS is an example of a proto-oncogene that, when mutated, will permanently activate cyclins to induce cell division at an exorbitantly high rate. There are other growth factor genes called tumor suppressor genes that normally inhibits cell division. So they normally put a brake on cell division. They normally, for example, the P53 gene is a tumor suppressor gene. Normally, P53 makes sure that all the errors have been repaired in DNA before it's allowed to go through mitosis and move into the cell cycle. And if a mutation would occur in a tumor suppressor gene such that their activity would switch off, then we don't have that suppression of the cell cycle like we should, and it can cause haywire and cancer as well. So cancer is essentially a failure of cell division to be controlled. unrestrained, uncontrolled cell growth, um, and tumors start to form. So the what is lost is the cell is no longer stopping at these checkpoints. It's no longer waiting to make sure the proper things are occurring before it divides, or it might not even wait if it might not even stop if there is no more room to divide. So that's kind of what happens with a tumor. It keeps growing and growing and growing, even though there's no room for it anymore. The P53 gene specifically, which was discovered at Stony Brook, um, plays a key role in the G1S restriction point. It halts cell division if it detects DNA damage, right? It can cause DNA repair. It can force the cell to go into G0 phase or keep it in G1 phase. Or if the damage is so bad, it could cause apoptosis or programmed cell death. All cancers at some point, all cancers have some shutdown of P53 activity. So all cancers have something common. They all have some shutdown of this P53 activity where it's not able to arrest cells at the G1S restriction point like it should. So here's a picture, a diagram about the normal operation of the P53 gene. So DNA is damaged and caused by heat radiation or chemicals. Cell division stops and P53 triggers enzymes to repair the damage, and then P53 trigger or P53 triggers the destruction of cells damaged beyond repair and allows cells with repaired DNA to divide. Now, in the abnormal case, when DNA damage has been caused and abnormal P53 or mutated P53 protein has been rendered inoperable, the P53 protein will fail to stop cell division and repair DNA, and the cell divides without repairing its damaged DNA. That damaged cell continues to divide, and if other damage accumulates, the cell can then turn cancerous. So the shutdown of P53 expedites the development of cancer by allowing mutations and damaged DNA to accumulate over successive generations. Cancer isn't just a single mutation. It usually develops after a cell experiences six or more Q mutations or hits. And cancerous cells have unlimited growth, so a turn on growth promoting genes. They ignore checkpoints, so like they turn off tumor suppressor genes like P53. They are able to escape apoptosis, so they don't go through normal programmed cell death. They are immortal, so they divide to an unlimited extent. So chromosome maintenance genes are turned on. Um, Cancer cells promote blood vessel growth, so cancers can grow their own blood vessels in their tissue. So they turn on blood vessel growth genes and they overcome anchorage and density dependence. So they shut off these touch sensor genes. So a cancer isn't just one thing got bad or one mutation, it's a host or a collection of key ons and offs, mutations that allow this aggressive form of out of control growth that we know of as cancer. Many different things can cause these hits or these mutations, like radiation, chemical exposure, heat, age, genetics, cigarette smoke, things like that. Breast cancer being one of the really important and common types of cancer. Here in a picture, we kind of I kind of show like the progression of that from a tumor that grows from a single cancerous shell and it invades neighboring tissue and lymph vessels and then can spread through lymph and blood vessels to other parts of the body. So this very localized single-celled something has gone wrong can then spread and spread and spread once it interacts with lymph or blood vessels that can spread it throughout the body. So a tumor in general is just a mass of abnormal cells. You can have a benign tumor, which is an abnormal cell that remains its original site and has a lump. So in this case, P53 has halted cell division. It's not getting bigger. And in this case, most don't cause any serious problems and they could be removed with surgery. You don't have to have chemotherapy and then you're done with it. Malignant tumors are the ones where the cells start to grow and leave their original site, and they lose attachment to nearby cells, and they may be carried by blood or lymph to other parts of the body or to tissues and start more tumors. And this is the process we call metastasis, and that's much more serious. And that's when we need very aggressive radiation and chemotherapy um, to treat things like that. That's kind of all I wanted to make sure to talk about for right now because in class we're going to go much more deeply into cancer and into the loss of control of the cell cycle and how those things play into one another.